So today we are going to discuss about the development of eye or development of eyeball. So as we have discussed in our previous session that different parts of the eyeball, what are the name of the different parts which is present in the eyeball. So we have cornea, which is present in the anterior portion, which covers the area from the foremost part. The second one is the sclera, which is the outermost covering of the eyeball. Then we have discussed about choroid, which is present just below the sclera, and then we have discussed about the retina, ciliary zonules and lens. So we have discussed different parts of the eyeball on our previous session. So today we are going to discuss about the development of eye, that how this picture or this parts of the eyeball comes into the position, that whatever we are learning here. So let's start from the very basics and from the embryology. So let me first make a diagram. Okay. So first of all, during the embryological period, or you can say during the gestation period, when there will be a formation of an eyeball, the first and foremost thing is the first stage, which is a formation of a neural plate. So this is a neural plate which is formed, and this is the surface ectoderm. So the first and foremost thing here in the embryological period is the neural plate formation. So this is the first stage during the gestational period. Then this neural plate will go into a process during the embryology which is called folding. So the folding process takes place here. This is the surface ectoderm and this is a neural plate. So the neural plate in the stage 2 during the embryological period, during the embryology, it will get fold like this in the folding process, and it will makes like a groove. And the third stage is the formation of neural tube. Neural tube. This is the stage 3. So actually what happens here, at the very beginning during the embryology, the formation of neural plate takes place along with the surface ectoderm, which goes into a folding process in which there will be a formation of a groove of the neural plate in the stage 2, and then finally it will form neural tube in the stage 3. So this is the first and foremost thing what actually happens. Now we will discuss further. Okay, so we have a neural tube. Neural tube. So the neural tube is having a two part. This is the upper part and this is the lower part. The upper portion of the neural tube is called caudalum and the lower portion is called caudiculateral. So the first foremost part of the neural tube is the phelicatinin, and the second portion, the lower part, is called caudalian. Now, the neural tube is going to a process in which there will be a formation of a 3 bulging areas of the neural tube. 3 bulging areas of the neural tube. So this is the first stage, and when it goes to a formation of a bulging, a popping out area in the upper portion of the neural tube, so this is called a forebrain. The second groove, the second bulge out area is called midbrain, and the third bulge out area is called a hindbrain. So in the upper portion of the neural tube, we have 3 bulgings in the stage 2. First one is the forebrain, the second one is the midbrain, and the last one is the hindbrain. So the first one is a prosencephalon, the second one is the mesencephalon, and the third one is the rhombencephalon. So these are the 3 different bulge out areas, pop out areas, which forms in the second stage of the neural tube, which is a part of a brain, which are forebrain, which is called prosencephalon, the midbrain, which is called mesencephalon, and the hindbrain, which is called a rhombencephalon. So now we will see how the eyeball is going to be formed. So we have 3 bulge out areas of the neural tube, which is a rhombencephalon, a mesencephalon, and a prosencephalon. So this prosencephalon is going to again bulging out into 2 different parts in a later stage. So the prosencephalon, this first part, prosencephalon, is going to be divided into 2 parts, and these 2 parts are called as, this is called as telencephalon, and this portion is called as diencephalon. Okay, so the first portion of the prosencephalon area which is divided is called telencephalon, and this area is called a diencephalon. So actually there is a bulging or a division of a prosencephalon area only in the stage 2. This is 1 and this is 2, in which it is divided into telencephalon and diencephalon. Okay, now the another very important thing here, what we need to understand, is the formation of an eyeball from the area which is known for source area is the diencephalon. This diencephalon area will go further bulge out in order to make optic vesicles. So this is an optic stalk, and this is a part of optic vesicle from the area of diencephalon, it is going to be moves further, form further. So this area, which is called as telencephalon, and this area, which is called a diencephalon, from here the formation of optic stalk take place to make an optic vesicle. And in this optic vesicle, the upper portion will form the pigmented epithelium of a retina, and the inner portion will form the neurosensory layer of retina further. So as we know that the development of actual eye starts from the area of diencephalon at the 3rd week of gestational period in order to make a formation of an optic stalk and the optic vesicle. So the outer portion of the area will form the outer retina, or you can say the pigmented layer of retina, and the inner portion of this will form the neurosensory retina. And the area here in the center will form the optic cup, this area. This area is the same area here after the formation, and we are just comparing with the formed eyeball and the developmental eyeball here, where this area will called as an optic cup later on. And this area is the same, this area which is called as the inner retina, and this upper area which is called as an outer retina. After this, there is a formation of choroid, there is a formation of sclera later on to form the outer covering, and again there is a formation of a cornea here in the front surface. And from the surface ectoderm, which is present initially, I have explained in the diagram. So surface ectoderm, from the surface ectoderm there is a formation of a lens placote, which further gives you a complete crystalline lens. And this gapping of this area will form the pupil, this one. This is the entrance area, this is the entrance point of light ray in the retina. So the area from where the light will pass is the aperture in between the iris, which is this area, which is called as pupil. So if you will compare this first diagram with this complete second diagram of an eyeball, then most of the thing you can correlate with this. That first and foremost thing is the eyeball is derived from the diencephalon area, in which there will be a formation of an optic stalk and the optic vesicles. After that, this area will form an optic cup. The area of the inner portion will form an inner retina. The outer portion will form an outer retina or pigmented area, after which the above areas are a choroid and the sclera, and the aperture in between will form a pupil later on with an formation of a lens here in the center part. So this is the formation of an eyeball, this is the formation of a retina, this is the formation of an actual formation of area from this portion to this portion. So you will better understand that how this picture of an eyeball, whatever we are seeing here, comes from or derived from the embryological period. Thank you.